Hello friends, welcome to my YouTube channel Dr. Sam's Biology. In this video, we will discuss molecular basis of complementary genes. We know that complementary genes are the genes that complement each other. So these are two genes that are present on separate loci. Means they are non-alleling genes. They will interact together and produce a phenotype. When any of them is absent, then the phenotype is not produced. So we will look into the molecular basis, how it happens. We know that in case of complementary gene, when we cross white flower sweet pea with white flower and they have genotype capital C, small p, small p, small c, capital P, we will get purple flower. But when we select them, we get 9:7. 9 are purple colored and 7 are white. So in this case, you can see that when both two genes, capital C and capital P, are present together, then they are colored. Color is purple. But when any of them is absent, then color is white. In that case, you can see that white because capital C is absent. Here, in that case, you can see that capital P is absent here, so color is white. So how it happens? This color, purple color of sweet pea is determined by a pigment that is called anthocyanin. And this synthesis of anthocyanin is a biochemical reaction and it has two steps. This reaction starts with precursor A. When capital C gene is present, then this gene will produce an enzyme, and this enzyme converts this precursor A into colorless intermediate. In second step, when gene capital P is present, it will produce an enzyme. This enzyme will convert this colorless intermediate into anthocyanin, and this anthocyanin gives purple color. So this is first case. When both these genes, capital C and capital P, are present, color is purple. Because this converts precursor into colorless intermediate and this colorless intermediate is converted by this capital P gene into anthocyanin. If any of them is absent, suppose this gene is absent, then this precursor can't be converted into this colorless intermediate. So if this colorless intermediate is not there, then this capital G, the enzyme produced by capital P gene, can't act on substrate because it has no substrate. So anthocyanin cannot be produced. So when both these genes are present, then anthocyanin is produced and color will be purple. In second case, precursor A is there. When gene capital C is absent, it is absent. Genotype is small c, small c. So if this is genotype, then there is no enzyme. So if there is no enzyme, then precursor A cannot be converted into colorless intermediate. So there is no intermediate. If there is no intermediate, then this gene, the gene capital P is there, but it will produce enzyme. Enzyme will be there, but it has no substrate, no colorless intermediate because here gene is absent that can convert this into this. So there will be no anthocyanin. So in case of color will be white. In third case, precursor A is there. Here capital C gene is present. So this capital C gene convert this precursor into colorless intermediate. It will produce enzyme. That enzyme will convert it into colorless intermediate. But here capital P gene is absent. So if capital P gene is absent, then there is no enzyme that can convert this colorless intermediate into anthocyanin. So if there is no enzyme because this gene is absent, then there will be no anthocyanin. If there is no anthocyanin, then color will be white. So in that case, capital C gene is present, but capital P is absent. So when both genes are present, because this reaction is catalyzed or completed in two steps. So when both enzyme for this enzyme and this enzyme is present, then color will be purple. Any of them is absent, then color will be white. So this anthocyanin production requires two dominant genes, capital C and capital P, and capital C and capital P both complement each other and produces purple color. So this is molecular basis of complementary genes.